Hi, my name is Aldo Guzman-Saenz from IBM Research, and today I'll talk to you about RubricOE and what machine learning can say about Alzheimer's disease. This is a collaboration with Daniel Platt, Filippo Utro, Aritra Bose, Subrata Saha, and Lakshmi Parida. Alzheimer's disease is notable for having substantial heritability unaccounted for by single nucleotide polymorphism alleles. A small handful of SNPs strongly linked with the ApoE E4 allele show very strong odds ratios associated with Alzheimer's. This study seeks to answer the question of what machine learning may reveal about pathogenic processes through identified alleles. Since we are working to predict values from extremely underdetermined features, we apply regularization and cross-validation techniques to exclude spurious predictions. We apply regularized linear regression to estimate heritability explained by SNPs. We score features by using models comprised of leading ranked SNPs with linear kernel SVMs to yield Uden scores. We contrast with predicted association obtained via logistic regression, GWAS style, and we also compare how often features were identified across multiple data splits, as well as the predictivity of selected sets by logistic regression versus stability counts using linear kernel SVMs. Data were acquired on June 26, 2021 from the Alzheimer's Disease Sequencing Project repository at National Institute of Aging Alzheimer's data storage site, comprised of data not excluded for use by commercial entities. We filtered more than 14,000 samples, retaining more than 78 million SNPs marked as passed by variant quality score recalibration in the GATK suite. Phenotype descriptions include age, sex, race, ethnicity, and ApoE4. In Alzheimer's disease, chromosome 19 contains ApoE, whose ApoE4 variant is very strongly associated with disease expression, with a number of variants in ApoE and nearby genes showing strong association likely boosted by linkage. We apply RubricOE to chromosome 19 and to chromosome 11, which has had 4 variants associated with it in large meta-studies. Alzheimer's is a challenging problem with relatively weak signals for everything except ApoE4. While performing preprocessing, we observed that the math histogram showed significant spikes. SNPs contributing to these spikes share identical allele frequencies and are often monoalleles with identical missingness across samples. This systematic missingness likely reflects batch variations where SNPs did not pass within subgroups. SNPs with high missingness were excluded. This removed the spurious spikes from the math histogram, and it also removed large numbers of very rare variants. After this, around 4.9 million SNPs were retained. Among the SNPs that did not pass were the ApoE haplotypes, though other ApoE and nearby genes SNPs survived. The idea for feature ranking is as follows. First, we consider a simple linear regression and identify explained heritability. Then, we apply L2 regularization, and then we identify the part of explained heritability corresponding to regression results. Here we go into a little bit more detail about the link between rich regression and heritability. The error for rich regression includes a regularization term. By introducing the B term and algebraic manipulation, we obtain a new description of the residual error. Thus, we obtain an expression of residual error that separates the impact of predictions of y from estimated x and the impact of regularization that does not derive directly from sampling variations. Additionally, we have a contribution from components of B to heritability which excludes impact of correlations, linkage, etc., and represents what is usually reported as heritability explained by that component. We use this to rank our features. After ranking, we construct score curves by considering multiple splits of working data as well as nested subsets of features. Each split gets a score from fitting a linear SVM and evaluating its performance with Uden index. Top features are selected by considering the set of features where the first local maximum along the curve was reached. Here we see that the first local maximum happens quite early in the curve, suggesting that the subsequent increase in Uden score corresponds to overfitting. To summarize what we've done so far, here we have an overview of the entire pipeline. Here on the left we have the full pipeline, starting from the initial data split into working dataset and validation, and on the right we have the structure of the main RubricOE loop. It is in this loop where the previously mentioned feature ranking and score optimization curves are computed. Zooming in a little bit more, On the left, we see that for each of the loop's repetitions, the ranking is done across multiple splits, and then an aggregated ranking is computed. On the right, we see that based on the aggregated ranking, we select increasing sets of features to construct an aggregated union score curve. Below, we see the relationship between stable SNPs and their p-values for chromosomes 11 and 19. Support, in green, means in how many iterations of the main loop a given feature appeared, and on red, we see the usual p-value associated to that feature. Chromosome 19 shows decreasing significance as support declined from a maximum of 50 with a rough correlation with p-value. For our final evaluation, linear kernel SVMs were used to predict AD diagnosis from 3 different sets: from all selected SNPs, from those appearing most frequently across splits, and from those that were the most significant. For chromosome 11, inclusion of more SNPs improved predictivity. The more significant and most supported SNPs predicted similarly. This suggests a possibility of overfitting. For chromosome 19, the most significant and most supported SNPs yielded very similar predictions and did as well as the full set of selected SNPs. Machine learning methods and pipelines, such as RubricOE, tend to present more candidates than GWAS significance-based approaches, yielding similar predictive power. With sufficient signal, both approaches will likely show some consensus. Thank you.